excuse me. One of the reasons that Evo-Devo is particularly interested in fish is because of a major transition from vertebrates that lived in the water to vertebrates that walked on land. And for years, decades, centuries, millennia, there was a missing link, one of many missing links, but one missing link was how, and creationists, of which I'm sort of a type, we can talk about theology and evolution and how to reconcile them if you want, after class, some other day. I've got a meeting right afterwards. But there was a missing link. You know, God must exist because how could, or God or some higher power, how could some higher power that's a creator exist or how could not a creator exist? How would evolution lead from water-dwelling organisms evolving to be able to exist on land? And go from breathing oxygen dissolved in water to miraculously all of a sudden breathing oxygen in the air and not needing gills but needing lungs, and all of these things that would have to happen at the same time to go from swimming in the water and breathing water to walking on the land and breathing air. And that's why about a decade ago, a fossil was found that some claim, including Neil Shubin, who was the scientist who discovered this species, or this fossil of this species, considers to be an important piece in that missing link argument, which is we never had a fossil or evidence of a transitory species, that is one that kind of looked like halfway between a fish and a tetrapod. And so the discovery of this fossil Tiktaalik in the mid-2000s, I think? Maybe late 2000s? Relative. Started getting people more interested again in the evolution of limbs. So that's what we're talking about in Sticklebacks for one of the two papers next week, is all about limb development. And one of the most important genes in limb development in mammals, and in fish, and in reptiles and other vertebrates, is PITX1. And this is just a picture showing a PITX1, the plus-minus heterozygote. So this is a mouse on the left has one good copy of the PITX1 gene and one mutant version. And so we can see what happens to a mouse when it only has half of the copies of that gene it's supposed to have. It's heterozygous. It's got a wild-type version, and its limbs all develop pretty much normally. They don't show a wild-type mouse here, but basically that heterozygous mouse is wild-type. But when you get rid of both copies of this important developmental gene, then the limbs, especially the hind limbs, barely develop and they're malformed, and the bones are different lengths other than what they normally are, and so it really screws up development. And that's an old paper that showed that that gene is particularly responsible for limb development, PITX1. And then there are also, but especially in this case, hind limb. So there are a lot of genes that have different roles. As you may appreciate, at some point you have to decide whether or not you're going to develop a leg where your arm is or an arm where your leg is. So that's one of the roles that PITX1 plays, is determining whether your forelimbs develop as forelimbs or hind limbs or not at all. Yeah?
So some of these like homologous structures, like in the whale fins, do we know, like, do they share these genes for these structures across these different species?
Yeah, in general, yes. The genes that control limb development and that's where we're headed, so that's a good segue into it, is that the same genes control development in fish and in terrestrial animals. And so we're going to see a little bit of evidence for that. We're not going to see a lot of it today, but that is the idea, is that what turns a fish's pectoral fin into a pectoral fin, these fins, are the same thing that develops as our arms. So our arms are homologous structures to fish pectoral fins, for example. And then the question is, what are our legs homologous to in fish? That is, what's the same structure? What's the thing that's created by the same genes in development in fish versus in terrestrial vertebrates? Another thing that's less important to know, but still relevant, is that there's another gene, PITX2. So there are two different genes, 1 and 2, kind of like there are multiple Hox genes, there are multiple PITX, well, there are 2 PITX genes. And one of the fates of redundant genes, or gene duplicates, is sometimes they can compensate for each other. If they duplicated recently, the two genes maybe do the same role. And we also talked at the end of one of the classes about what other possibilities are for duplicate genes. They can do the same role, that's redundant. And then there was subfunctionalization. So if each gene has two functions, then this gene could keep function 1 and that gene could keep function 2, so that in whole, both of those genes are needed to do all of the roles of the first gene. There was also subfunctionalization, neofunctionalization. There was a third option. So in this case, this PITX2 copy of PITX1 gene is partially redundant. And the reason that they know that is from the same paper is that these heterozygotes, you can see the right femur in the knockout, minus minus, is really tiny. What happens on the left side, though? It's longer. So even in the absence of PITX1, the PITX2 gene is still able to do some femur development. But the amount of femur development it does depends on which side of the animal you're looking at, which is odd. And this is not just one-off. They've proven that that's the case, that PITX2 can do femur development, but it does it better on one side of the mouse than the other. So we talked a lot about symmetry and patterning in terms of anterior-posterior in this class, Hox genes. Tell which part of the body is developing as anterior, thorax, abdomen, and so forth. And what we haven't talked about so much are the two other axes of development, dorsal ventral, stomach to back. There are another set of genes that control what parts of your body develop more as the stomach side and the back side. But in developmental biology, what's known least well is how left-right asymmetries form. We are definitely asymmetric head to tail. We're definitely symmetric stomach to back, dorsal ventral, but not so much left to right. But there are some examples here of really important left-right differences in development, mostly organ systems. We've only got one heart. It's not in the middle of our body. It's on one side. So it normally develops on the left side of our body. So this is a really interesting puzzle for developmental biologists, and it's been really hard to figure out what genes control those left-right asymmetries. So this is just showing examples from the gut, the intestine, the heart, and the lung. Most of our internal structures, internal organs, have some asymmetry to them. And so very few genes are known. But it turns out that PITX2 is one of these very few genes that's known in many cases of asymmetry to play an important role in having development occur more on one side of the body than the other. Which I think is fascinating, but that's because I've got a left-handed daughter, which probably has nothing to do with PITX2. That's not development, that's probably neurobiology, not so much development. Okay, so limb development, specifically in fish, because we're talking about fish. So at the base of this tree, we've got chondrichthyes, which are the chondrocyte or cartilage-containing fishes, so the non-bony fishes like sharks. They diverged first, and we've got osteichthyes, which is ostei-bone, the bony fishes. And so they all share a common ancestor, and then what we're trying to get to is fish that are related to animals. So we've got the next important split is actinopterygii. Don't remember this stuff, I'm just throwing out fancy words in case you never knew how to pronounce them but want to know how to pronounce them. The actinopterygii are the ray-finned fishes. So most of the fish you think of as fish are those ray-finned fishes. They have, especially pectoral fins, you can see lots of sort of bony-ish structures in the fins and in the tail as well, the dorsal and the caudal fin. They all have rays in them. So those are ray-finned fishes. That's like 200,000 species or something. It's tons, especially if you're an aquarist or in the trade, which I'm not. You know there are a lot of different fish species. But then we get into the weird and very relatively rare fish, the sarcopterygians, which instead of ray-finned fishes are called lobe-finned fishes. Like earlobe type lobe. Those are the sarcopterygians, or the lobe-finned fishes. They're more closely related to us. They shared a common ancestor more recently than we do to ray-finned fishes. So this is the trajectory leading to, at the upper right there, terrestrial mammals. So one of the oldest versions of a lobe-finned fish is called the coelacanth, which was another missing link type fish that was discovered in a fish market in Madagascar. Like, the local fishermen knew about it. They'd been catching them from the deep for years. They just didn't realize the biological importance of them. But at some point, as I understand it, a developmental biologist was in Madagascar, and went to the market and said, "Oh shit, what is this?" It's a lobe-finned fish. They're super rare, but the fishermen were like, "Oh yeah, we catch those every once in a while." So they had this specimen then they could study and understand better the evolution of vertebrates with that fish. And then there are also some funny fish that are sarcopterygians, like the lungfish, which can breathe air. So this is another kind of missing link fish in that it can sustain itself for a short while outside of water. These are the ones, well, there are also some lobe-finned fishes that are able to actually walk. They use their flippers, but they can leave water and they can across the land and get to another body of water. They're hugely invasive. Trying to remember the name of the fish. I just saw them in the last month or so, some governmental organization in the East Coast or the Midwest, snakeheads. Whenever they show up somewhere, there's always this huge clamor about, if you see it, kill it, because they're, not only can they do that, they can survive out of water for a short period of time, so they can move in between bodies of water, but they like devour everything that's in the water. They're top predators. So anyway, for what it's worth, those are the types of fish that are close relatives of ours that are still considered fish. And the most recent one then is that Tiktaalik, the missing link fish, or some have called it the fishopod, because it's a fish, but it's got things that are starting to look like developing limbs. Its limbs are a little bit longer and a little bit bonier than ray-finned fish and even lobe-finned fish. And that was thought to be, again, this sort of missing link idea of, if you're going to be terrestrial and you're going to be able to support all of your weight on your limbs, or some of your weight on your limbs, you need bonier, heavier bones in your fins to be able to support your weight on land. And then there was the evolution from a close ancestor of those fish, then terrestrial organisms evolved. So how does that have to do with Threespine Sticklebacks? Well, that's just the background in sort of limb development. So these are small fish that are found all around the world, except in the tropics. And they're found both in marine and freshwater habitats, which is interesting for other people, especially physiologists, trying to figure out how these fish, some populations of these are anadromous. They're like salmon, they go out into the freshwater to mature, and then they return to freshwater to spawn. And how they can cope with high salt water and then freshwater is interesting as well, but not part of Evo-Devo this term. And there's a big difference between those marine and freshwater fish. So what these images are, they're alizarin red stains, which stains bone, basically. And what this has shown is that these Pacific marine fish, these fish don't have scales. Instead, they develop these bony plates that are kind of like armor. So they're underneath the skin. They're not on top of the skin like scales are. They're underneath the skin. But presumably, so all of this red here are these bony plates that protect the internal organs of Threespine stickleback marine fish. So they have these bony plates, and they also have their three spine sticklebacks, first, second, and third dorsal spine, right before the dorsal fin. So that's a fin, but that's actually not part of the fin. That's its third spine. I'm not surprised if you overlook it. But what happens when these marine fish have colonized freshwater habitats? What happened to all that protection? What went away in the Paxton Lake population?
The armor?
Plates got evolved away.
The pelvic spine.
Pelvic spines are gone, and also dorsal spines got severely reduced in size. And there are lots of reasons. So the loss of these plates has been studied separately from what we're talking about next week, which is loss of the pelvic spines. So this is the specific trait we're going to be talking about in the paper next week, one of the papers next week, is these pelvic spines. Not the dorsal spines, but just that bit. And we know from other studies that we're not going to talk about, but are also good Evo-Devo studies, what genetic changes cause the loss of the bony plates. That's ectodisplacin, which is a gene we've seen before in class. We talked about it really briefly a couple of weeks ago. Anyway, don't need to go there. So that's what causes loss of the bony plates. But what was unknown at the time of this paper was what genetic changes causes the evolution of loss of the pelvic spines. So this is not only a big developmental transition in that it's loss of an appendage, but it also happened in a really short time on an evolutionary timescale, like 10,000 years or less. So PITX1, again, is involved in limb development. Pectoral fins are the forelimbs of the fish. These pelvic spines, well, I don't know, do your legs grow out of your pelvises, more or less? Femurs, pelvis, right? Pelvic spines are the fish's hind limbs. Those are its leg equivalents. So basically, in Paxton Lake, the fish lost their legs. That's the generic way of saying it. And so the scientists that discovered that initially thought, well, okay, let's look at the PITX1 gene because we know it's involved in limb development. They referenced that mouse paper that showed that if you delete PITX1 or mutate it, that the mice don't develop their hind limbs at all, as we saw in one of those pictures. Hind limbs are totally deformed, their bones are really short or missing. But they found that there was no coding difference. That is, the protein produced by PITX1 is the same in the marine form, like on top, and the freshwater form on the bottom. So it wasn't that there was a difference in the protein sequence of PITX1 that causes this difference. So what does that mean? What's the likely genetic basis of a change in evolution if it's not a change in the protein itself?
Non-exonic region.
Conserved CNEE. And
Transcription.
Yeah.
right, so it could be that the gene is fine, but somehow it's either not being transcribed or it's not being something properly. So there's no mutation in the gene, there's probably mutation outside of the gene. We've seen that before at least a few times in this class. And they also discovered that by study, not discovered. They looked for and discovered that this gene is still expressed in most of the body, which is normal, but it's not, in this fish, it's not expressed in the pelvis region. That is, this not only is the gene present and the protein sequence is fine, but it's expressed in most of the regions of the body where PITX1 is normally expressed. It's just absent in one particular spot in the body, and that's the pelvis. That is, the gene's in the pelvis, but it's not turned on in the pelvis. It's not expressed there. And so that set of evidence suggested the bottom bullet point, which you've already read probably 5 minutes ago, so we can move on. Suggests that there's a mutation that causes, specifically in the pelvis, PITX1 not to be produced, and therefore doesn't develop hind limbs or pelvic spines. So here's where it gets really weird, and I hope if you find anything interesting, it's going to be this slide and the next one. Don't skip ahead. I want to save the surprise for just a minute. So we've already talked about the fact that these deletions and relying on PITX2 causes an asymmetry in hind limb development, where the left side is usually longer than the right side. The same thing is true in these spineless stickleback and spiny stickleback populations. The left side is usually longer than the right side, the left spine. And that's getting back to the question about, is there consistency in the genes that are used in fish and in terrestrial mammals to do the same sort of developmental process? Yes, absolutely. We even see the same sorts of odd patterns like that from the same genes in fish. and, drumroll please, the same genes in terrestrial animals. This really bizarre paper in 2006, I mean, it's not bizarre, it was shocking in a good way, shockingly pleasing. So Mike Shapiro and others who are stickleback researchers were interested in that thing about there's this weird asymmetry when PITX2 controls limb development that the left side is more often longer than the right side. It's true not only in sticklebacks, in three-spine sticklebacks from different environments and also the nine-spine stickleback, a different species, but in elephants and manatees. So they got data, morphological data from manatees in Florida and discovered that in those manatees they also show the same difference. If a manatee's hind limbs, which basically don't develop, but there are still rudiments in manatees. So they went, I think Mike they flew to Florida and like examined a bunch of manatee skeletons or something, took measurements of them, and found the exact same thing, that if there was any difference, percent excess length left versus right side, just comparing the left and the right vestiges of the hind limbs in manatees, most of the manatees had left side longer than right side. Percent excess, left side. So anything underneath 0 would be they were longer on the right side. So there were very few manatees that had longer right hind limbs than left. But a whopping majority of them had some of them had really longer left side hind limbs than right. And almost every skeleton they looked at, or manatee they looked at, did have a excess left length over right length. So this is a thing. PITX1 and the PITX genes control limb development, and they tend to make the left side longer than the right side for whatever reason.
So, the PITX genes,
Yeah.
I'm searching Google, the homeobox genes, what are
Right, so PITX stands for pituitary homeobox. They're homeobox genes, which generally means transcription factor. The genes that have homeobox, a homeobox is a domain, it's a part of a gene, and genes that have homeoboxes tend to be, or homeodomains, tend to be transcription factors. So it's a transcription factor that responds to pituitary hormone. That's what the PIT, yeah, that's what the PIT part is from. They respond to pituitary hormones. Yeah. So sticklebacks have left-right asymmetries, so do elephants and manatees, and presumably humans too. Okay. So PITX genes, sticklebacks. Here's where sticklebacks live, all the red shaded areas. They were ancestrally marine, but let me see if I can get this straight because I'm not a geologist or a hydrogeologist. Is that a thing, hydrogeology? During the last ice age, the ice sheets, correct me if I'm wrong guys, if you know this, the ice sheets weighed down the land, so sea level was relatively higher. What we consider now land, especially on the coast, was underwater because the ice sheets were weighing down on the land and pushing it down. And as am I right? Am I close? I don't know. This is what I heard. That when the glaciers...
That sort of sounds like a lot of mixed things.
Okay.
Like two different things. It's more of a, there was more water trapped in ice, so.
Okay.
And there was
Okay.
water pushing down in the ice, but that was more of a gravity thing. So I don't know.
Okay.
Describe it. You said.
Well, let me finish and then tell me if I misstated something to your knowledge. So what I heard was that what's currently coastline, which are basically most of these red shaded areas, were submerged at the last glacial maximum. And as the ice sheets retreated and melted, the land lifted for whatever reason. So I won't say why, but the land that had been submerged lifted up a little bit. And what that did was it stranded a lot of, this part's true, I don't know about the last part, a lot of marine sticklebacks were stranded and trapped in regions that became lakes. So they were previously submerged, so marine fish were swimming everywhere, didn't matter where they were because they were all underwater in the ocean. But when the land raised, at some point a lot of stickleback populations got trapped in these lakes near the coastline. And so what's really cool about sticklebacks is that there are literally hundreds, probably more, of lakes around the world where that marine ancestral population got trapped and were reproductively isolated from all the other fish. That is, there were like 40 fish in this lake, and they've lived there for the last when was the last glacial maximum? It was like 4,000, 3,500?
25,000.
25,000. So in the last, yeah, thank you. In the last, that time scale, all of these different stickleback populations have been evolving and growing and reproducing and mating independently of each other. So there's this really cool naturally occurring experiment, basically. We had the same ancestral population and got put there. They can't get out, so they can't mate with each other. So it's just a question of where will you put, what are the environmental conditions, and how do those sticklebacks evolve to adapt to those different freshwater environments? Thank you for the details, I appreciate it. So the next slides are just examples of some of the places these sticklebacks are found, especially in the Pacific Northwest. So here's Vancouver, Canada, not Washington, and Texada Island. Texada Island is home to Priest Lake, this little tiny lake, which is pretty close to the ocean, but not connected to it. No streams out or in. There's no way for sticklebacks to get in there unless somebody puts them in there or takes them out. So that's one population that we're going to read about next week. So it's a Canadian stickleback population, one of these isolated lakes. Another one is Bear Paw Lake, which is just inland from Anchorage. Check this out. It's got a review. You know you were only here because of science. I don't know who this person is, but I'm going to try to find out, because there are a lot of people that work on sticklebacks, but it's still a pretty small community. But it got a 5-star review. So there's Bear Paw Lake. We've got the Little Camel River, which isn't a huge river, but it's in almost downtown Vancouver, British Columbia again. That's another population you're going to hear about. Hump Lake, which is also near Anchorage, but just to the southwest, another isolated population. So those are the populations you're going to hear about in this paper. That's where those places are in the world. They're all in the Northwest. So the question is, what happened to all these marine sticklebacks as they invaded all these freshwater populations? What came out later? Well, one of the things that happened was that the predators changed, because they were no longer in open water where these ginormous fish that sticklebacks are about that long, about that size. So a salmon could swallow one, a trout could swallow one, lots of marine fish could swallow them. And so the marine ancestral populations are the ones that have all the spines, the body armor. Presumably, if a fish tried to eat them, what they do is they stick out all their spines, and the bony armor protects them from being bitten, and then the spines maybe get lodged in the big fish's mouth, and the big fish tries to spit the stickleback out or something like that. But when they looked in freshwater, there are different predators there. There aren't these big fish living in these tiny lakes. They actually, one of the predators, aside from fish, or aside from birds rather, are dragonfly larvae. So this is an example of sticklebacks doing their defensive thing when being attacked by different predators. So you can see in each of these cases, maybe you can see, that the dorsal spines are spread way out when they get attacked. And you can do this in the lab too when they're swimming in an aquarium. You like tap on the glass or something, fish freak out.
I don't think it helps, it's helping them.
No, it's not. Well, it does, because the bird, see, it sticks out its dorsal spines. It's 3 dorsal spines and it's 2 pelvic spines, and then the fish can't actually get it down its throat. It hurts, right? If the bird tries to swallow this spiky thing. The same, this tossing the stickleback in its mouth, I guess, but its spines are out. But then there are the dragonfly larvae on the bottom left. They're not trying to eat the stickleback whole, and they don't really care about the spines. And in fact, well, maybe not in fact, story, the idea is that those dragonfly larvae actually predate the sticklebacks by grabbing their dorsal spine, or their pelvic spines. That is, it's a grabbing onto point for certain predators. That's one of the theories why a lot of these fish in the freshwater lakes stopped having pelvis. That is, they evolved lack of pelvis and lack of pelvic spines, maybe because it's an adaptation to the types of predators that are in those lakes.
And they're also being selected out in that instance, right?
That, exactly. So if you were the rare fish that at some point in the evolution of the fish in that lake got a mutation that would cause you to lose your pelvis, you'd be at a substantial advantage. Everybody else, your brothers and sisters and cousins, get predated, and then your lineage lives on. That's a story. There's some data to back it up, but it's a hypothesis.
So in
That
the,
predation is why there's a loss of pelvis. Yeah, it's
in
fine.
the freshwater environments, they're missing pelvis, but in the other environments, they're not.
Correct.
Okay.
Yes. So the ancestral marine population, which is just one population, go out into the Pacific Ocean, collect sticklebacks. They're fully plated and they have pelvic spines. Most freshwater environments that have been looked at, the fish, those independent evolutionary studies where fish got trapped in isolated lakes, most of those populations have lost their pelvic spines. Most sticklebacks don't have, most three-spine sticklebacks, especially in freshwater, don't have pelvic spines. And that means that they would have all evolved the loss of them independently.
In the marine environment, are they 100% with spines, with that pelvic?
There is some variation in the marine population. So not every marine individual is perfectly fully plated and fully pelvic, fully pelvised. I don't know if can I turn pelvis into a verb? I just did. So there is some genetic variation. And presumably, so that's one of the other debates that people had was when marine fish got trapped in these lakes, are we looking at there's two differences here, evolutionarily speaking. There could be new mutations that arise, which is what I just characterized. If you were the one lucky fish in the lake that got loss of pelvis, then you would be at an advantage. Or what was happening was, say 100 fish got trapped in the lake. Some of them have a little bit less pelvic spine than others, because there is natural variation. So maybe over time, those ancestral fish that already didn't have as much pelvic spine length got selected for. So it's one of those two. It's either existing mutations that got trapped in the lake, or it's new mutations in the lake. And there are lots of other hypotheses, which some of us, did we talk about that last week? Some of us talked about this briefly last week after class, I think, other reasons why these traits might evolve in freshwater. Aside from predation, there are other hypotheses for why you might lose the armor plating. That's what we talked about last week, after class. So if you didn't catch that conversation, if you're interested, I will repeat it now, or to you independently after class. But we're not really focusing on the bony armor, we're focusing on the pelvis this week. Okay, questions, thoughts, concerns? So basically this is a picture of a marine fish on the top and random freshwater fish from most of those lakes on the bottom.
What are like the closest relatives to these stick?
You're just poking my buttons. I'll talk about this for, what time is it? We end at 3:15, right?
Yes.
Seahorses are some of the most closely related fish to sticklebacks that you're familiar with, that I'm familiar with too. Yeah, that's the most closely related, commonly known fish to sticklebacks, seahorses. And it's partly because, do we have 3 minutes? It's partly because male sticklebacks do the parental care. They build nests, they try to attract females to lay their eggs in his nest, and then he swims into the nest and fertilizes the eggs. And then the male fish aerates the eggs until they hatch. So he keeps swimming into his nest and fanning the eggs with his fins to keep them aerated so they develop properly, and then they hatch, and then they go off. And seahorses are a classic example of paternal care. And they're close, so presumably one of the reasons they're closely related is because both of these types of species does parental care. The dad takes care of the kids, not the moms. Seahorses, of course, the dad keeps the embryos in his brood pouch, which is a sac, kind of like a kangaroo. So the mom deposits the eggs in the dad's brood pouch, and then the dad takes care of the eggs until they hatch. Yeah, it's a thing. So, good question. Okay, so here's your reference table for those populations and their pelvis situation. So complete means they've got a full pelvis and pelvic spines. Reduced and low are subtle differences, but basically that means they're not there. So we've got a set of fish. So the Japan Marine are that ancestral form, the marine fish. And then you'll notice that there's sort of replication. Most of the freshwater lakes don't have pelvis except for Little Campbell, that's a river, so that connects to the ocean, so that may be, I'm not sure if that's actually considered a freshwater population. Same with Salmon River. So we've got replicates. We've got multiple populations that have pelvis or don't have pelvis. Which one's definitely not saltwater that does have a complete pelvis?
Wait, what? What was the question?
So, which ones have complete pelvis? Salmon River, Japan Marine, Little Campbell, those are all, so that's ancestral. Those are the ocean-going Japan Marine fish. Those are ocean fish you catch today in the ocean. Salmon River and Little Campbell River are completely pelvised, but they're also pretty closely related to the Japan Marine ancestor. But the only other one on this list that's complete is definitely not a saltwater fish, because it's in Friant, like here, Friant. So there's a classic population of sticklebacks. You can go up to the Friant River or Friant into the river and collect them. You can see them swimming around. There is this really odd population that's been studied since the 70s that still have their pelvis but lost their plating, which is the only time that's been seen. So they lost some of their defenses, but not all their defenses, and so that's historically been it. Yeah, like just up the road, like 5 miles. Drive up there, stare in the river, look for some sticklebacks. Someday I hope to get a permit so I can collect some and study them.
Are they native or introduced?
They're native. Yeah. It's about as far south as sticklebacks get too, which is also interesting. So they will also be talking about the Friant population in this paper.
You mean like
And here's one example of a study from a while ago, so you can see that Friant population. I'm sorry, I got it backwards. They lost their plating, but they kept their spines. No, I said that. That's what I said, right? They're partially plated. They don't have all of the armor plates, but they still have their pelvic spines. So it's a complete population, but there's, again, that Paxton benthic fish with no spines, no pelvic spines, and, well, actually, one armor plate, just one little strip. Doesn't do much for the fish. But this is what geneticists is doing. So the reason I'm showing this paper, this figure, is because this is something that they're going to talk about, this type of experiment, in one of the papers for next week. Is that they cross individuals that, say, a marine individual that's fully plated and fully pelvised with a Paxton fish that does not have a pelvis. And they want to know how many of the offspring have pelvises. So if you took a fish that has a pelvis and a fish that doesn't have a pelvis, and you mate them together, no pelvic spines that is, full pelvic spines, no pelvic spines, and you cross them, what do their kids look like, do you think, in terms of pelvis development? So there may be 3 options. That one option is maybe they all have complete pelvis. Pelvis spines, pelvic spines, pelvis, the whole works. What are the other 2 options? None of them have any pelvic spines.
Or they have reduced?
Or they have either. So for this paper, they're just looking at total absence or total presence. There is a thing called reduced, but that's a subtlety. So for this, they're just going to score the fish as either they've got pelvic spines or they don't, for the most part. It's binary. So either all the fish have pelvic spines, none of the fish have pelvic spines, or there's a mix. Some of the offspring have pelvic spines, some don't. In other words, if spines is dominant, that is, if the ability to produce spines pelvic spines is a dominant trait, and you have, let's say that's, I don't want to use capital S and lowercase s, P, pelvis. So capital P will be normal wild type pelvis with pelvic spines. So that would be the genotype of a complete, presumably, an ancestor. Marine ancestral, it's homozygous for whatever gene controls pelvic spine development. You made it to an individual from a pelvic spineless population. Then you get, every individual is got one of each type of gene, a capital P and a lowercase p. They inherit one from each parent. So that's really the question is that genotype, capital P over lowercase p, going to produce spines or is it going to not produce spines? And if it produces spines, that would be because if they all get spines, it would be because that spine-producing version of the gene, the capital P, is dominant. It doesn't matter what you have with the other gene, that one version of the gene that produces spines is enough to produce kids that have spines, dorsal spines, or dorsal spines, pelvic spines.
Is there a reason why they turn into like the baleen whales, like the vestigial pelvics of baleen whales? Like, why do they just become these
Probably just disappear altogether.
vestigial structures? Like how whales have that pelvic structures, but they don't have these.
Right. Probably a different mutation that causes different loss of pelvis, or different degrees of loss of pelvis. I don't know. It's a good question.
Is there like what is that test they do, like a molecular test to determine if it's something is like has incomplete dominance?
Those are almost always cross-based, and that is sort of what this is testing, is what is the dominance. So if you saw all of the kids' offspring producing pelvic spines, then you would conclude that the capital P was dominant. If not having pelvic spines was dominant, then that lowercase p would be the dominant allele if you observed no pelvic spines in the kids.
So there isn't like a
There isn't like a molecular test like extract DNA or anything like that, unfortunately. That would be easy. That would be nice. Okay, and then we talked about cis and trans mutations before. So how does the answer to this question depend on whether or not whether a cis mutation or a trans mutation causes reduced pelvis development? I mean, we already kind of know the answers to one of these because we talked about it earlier today. So let's say here's one F1 individual. We're just diagramming one chromosome pair. And let's say there's a transcription factor gene that's important for turning on PITX1. Or, well, I gave it away. But we probably already knew I gave it away because we were talking about PITX1 so much at the start of class. Let's say there's a gene called the complete gene that's necessary to be turned on to produce a complete pelvis and spines. And there's a transcription factor gene that's important to producing the transcript. Or, yeah, we'll call it like that. So there's two genes, a transcription factor gene that turns on the complete gene. So if this is a cis mutation, what would that look like? If a cis mutation causes loss of pelvis development. Cis mutation means it's on the same chromosome as the gene that causes the trait. So what sorts of cis mutations could occur to cause? So there are two different types of mutations. Well, there's one particular type of mutation I'm aiming for, which is if there's a transcription factor binding site upstream of that gene. This is consistent with what they found. They said that the PITX coding region hasn't changed. That is, this is the same, this and all the other fishbowl diagram are going to have the same sequence of the gene. Whatever the gene is, PITX1. whatever the gene is that causes development of complete pelvis, the coding region sequence, the protein it produces is identical. So that leaves us with these two cis or trans opportunities. So this is a type of cis mutation that would block that transcription factor from binding the DNA and turning on transcription. So you'd get no transcription and you'd get no pelvis. And this, by the way, so this is an F1, which means that the top version of the chromosome, I failed to mention this, came from the complete parent. Oops, sorry, bottom chromosome came from the complete parent because it doesn't have a mutation that prevents the complete gene from being turned on. This top one is from the absent population. In other words, that bottom chromosome is the capital P. Is that how we defined it? Yeah, capital P was the complete population. So that top chromosome came from the lowercase p population. So that's a gene in an that's a chromosome in an F1 that came from the population that doesn't produce pelvic spines, and that's because there's a mutation that prevents a transcription factor for turning on transcription. The complete population doesn't have that mutation, so it can still transcribe that version of the complete gene. So that individual, if this exists, this hypothetical, if this individual existed, then they would probably produce a pelvis because there's one wild type copy of this gene that can be transcribed. Transcription factor could bind that version, one of the two chromosomes, and turn on transcription of complete. That's a cis mutation. What about the trans mutation? So trans mutation absolutely is when a mutation on another chromosome, or
Could it even be like on the same one, but like super far away?
well, we're gonna see how this works.
And, like, it
Absolutely.
turns
So in the trans situation, a trans effect is a mutation of something that's freely diffusible. So it's not a mutation as here, cis, that's linked, that is physically connected to one or the other or both of the genes. A transmutation would be an example. What should I mutate to cause this F1 individual to have or not have pelvis? We know it's not a mutation in the gene, complete. They're identical. We know it's not a cis, we know it's not the cis mutation, which is a mutation in a transcription factor binding site. So what's left? To mutate that would cause complete the gene not to be transcribed.
Transcription factor itself.
Yeah, the transcription factor. So if we had, again, the mutant version, let me choose blue instead of black. If we had the mutant version of a transcription factor, so that again would be the lowercase p, the absent population would have had two versions of that chromosome, the chromosome on top. So if you were homozygous for this top chromosome, then would you produce any transcription factor that could bind its binding site and turn on transcription? Because both versions of that transcription factor would be mutated. So in this hypothetical case, you get no transcription. And that's a trans effect because that transcription factor can't work on that gene, and it also can't work on, in trans, it can't act on the other version of the same gene that came from the other parent in the context of the F1 cell. But that doesn't mean that this F1 organism has no pelvis, because what does it have working at its advantage? Well, it's not dominant, but it's produced. So in this case, it is dominant to a mutation. So that transcription factor still exists, and it can bind to that chromosome and to that chromosome and turn on transcription of both copies of the complete gene. So in other words, when you do that sort of hybrid cross and you look at the offspring, you can, and this is what they do next week, which is why we're going through it in class, so hopefully this helps you understand how that analysis of their experiment works. You can try to distinguish whether or not they're cis mutations or transmutations based on the phenotypes of the offspring. So if you make F1s and there's a trans effect, and F1 should have a wild type version of the whatever the diffusible factor is, the transcription factor, should be enough to produce production of all of the whatever gene this is to produce a pelvis. So you should see pelvis. And you should see less expression of this gene, half as much, if there's a cis mutation, because that version, one of the two copies of the gene, will never be transcribed. And that's going to be the key difference between these two possibilities. You make F1 hybrids and you figure out what the ratio of, basically what the ratio of transcription is.
I have a question.
Yeah, please.
For trans, I know you said for cis, it's linked, so the genes are on the same chromosome? Is that,
The mutation's on the same chromosome as the gene that it affects, yeah.
that's different for trans, so trans can be, you can, if that transcription factor on, that lowercase p, if there's a mutation on that transcription factor itself, but that transcription factor on the dominant, or the uppercase p,
Right.
on the lower part, that can turn on the gene on another chromosome.
Right.
Oh.
Yeah, that's the idea. Or the other way to put it is if this was a dominant negative mutation, that is if that made this transcription factor go haywire and screw up the cell, then that would still be a trans effect. It would be that gene, that mutation is not just having an effect on the chromosome it's on and the gene that it's associated with physically, that mutation would also be having effects on other chromosomes. Hence trans, a cross from one chromosome to the other. In this case, it's a beneficial trans effect that transcription factor acts not only on its chromosome but also other chromosomes that have its binding site. So they're going to use this sort of a cross again, although they're going to look at F2s, not just stop at the F1s. They have to get to the F2, the grandchild generation. And what they're going to look for is situations where the F2 individuals have something common genetically. So let's say these were different genotypes. Now I'm using color coding, so I'll explain the color coding in just a second. So let's see, these are 4 different genotypes of 4 different fish. So 4 different chromosomes, and let me tell you what the phenotypes are. So those are the genotypes. And we're going to say blue is from the capital P, or wild type, pelvis producing population, and the red versions of the chromosomes are going to be from the pelvisless population. And the phenotypes of the fish are, this fish has a pelvis. This second chromosome was from a fish that has a pelvis, and the third and the fourth ones are from fish that don't develop pelvis. And we've gone through this, I think, once in class, sort of in theory, as how genetic mapping works. That was with the hairy leaves, if you remember. Like a month ago or so, we had a genetic mapping, hairy leaves, not hairy leaves, little exercise. So what are we looking for to figure out what region of the genome controls absence of pelvis? Should the region of the genome that controls absence of pelvis be red or blue? It should be red because the red chromosomes are from the fish that don't have pelvis. So we're looking for a region of the chromosome that has red. And on top of that, the other important thing,
in the middle
In
of
the position.
It has to be, there's a consistency we have to look for.
a little section in the middle.
It has to be, if there's only one gene involved, it will be the same region of the genome in every fish that doesn't have a pelvis. Which means we're looking at that little bit of the chromosome that's red in all of the fish. It's got that part of the chromosome. It's chromosome from a pelvisless fish, and that bit of the chromosome is in every one of the pelvisless fish. That's how genetic mapping works. Collect a lot of offspring, genotype them all to hell. That is, you get all their DNA sequence as much as you can, and then you look for that correlation. We're looking for a trait of no pelvis. We look for the DNA that came from the population of fish that had no pelvis and where it wound up as associated with phenotypes of their grandkids in this case. That's genetic mapping. You're looking for something that's in common among all of the fish, or as many of the fish as possible that don't have pelvis. See a little bit of that too in that paper. Okay. But so that is one of two different types of mapping experiments. That is collect fish from the wild, force them to mate in the laboratory, collect a bunch of their grandkids, and then extract DNA and sequence. And then do that computational analysis. Look for correlations of genotype and phenotype. This naturally occurring evolutionary experiment that Mother Nature did for us, though, is cool because there were so many different lakes, each of which independently evolved loss of pelvis, that you could use the same argument without doing all of the having to do crosses in the lab and collecting fish that way. You could just go out in nature into all of these different lakes, collect a handful of fish from each of the lakes, and assume the exact same thing. If the same region of the genome controls loss of pelvis, when you collect a bunch of pelvisless fish from all over the world, you should see the exact same thing. That there should be one region of the genome that's consistent in all of those fish, no matter where they're from. If they've got this trait, if there's only one region of the genome that you can mutate to cause that trait, you'll see that. So that would be same sort of diagram. Collect a fish from Bear Paw Lake and its ancestral marine, for example. Alleles are blue. And if you get one from the Little Campbell River, and if you get one from Hump Lake, something like that, then you still have this same region of consistency between all of the independent populations that have each lost, in its own way, pelvis. Because that's a different type of genetic mapping called association mapping. Which they will use. Transgenesis, another thing they're going to do. This is an experiment they're going to do. They're going to take the promoter of PITX1, and they're going to put it in a construct upstream of the GFP gene. What would happen to a fish if you put that DNA construct in?
If they have the PITX1 protein being expressed, then they will become green.
Right. So if you put this in a fish that has whatever transcription factor it is turns on PITX1. You're right, that's the correct logic. It's not PITX1 that binds its own promoter, but whatever transcription factor turns on PITX1. If that's present, then you should see green fish. And what would that suggest about the fish you injected that construct in?
That they have pelvis? No?
Well, at least they have the transcription factor that is used to turn on PITX1. They've got a sequence, they've got whatever protein, that's the trans factor. We haven't introduced that into the fish. The fish are using their own, whatever fish got injected already has this transcription factor. That's why we would see green fluorescent protein produced. So that's basically to check whether or not the PITX1 promoter of the complete fish is enough to turn on transcription. So they're going to check the promoter regions to see if they're able to produce transcription factor. What happens if you take the PIDX1 promoter and gene together from a complete fish, so a fish that does have pelvis? What happens if you take that DNA and put it in a pelvisless fish? What's going to happen if you inject that into an embryo? What do you think will happen? An embryo from a no pelvis producing population, you put just this bit of DNA in it.
You should start expressing that X1 protein. No?
If it has the transcription factor, it should have pulses.
Right. So you're both right. Yeah. If this no pelvis producing fish has the transcription factor it would need, if it had this gene, then it would have a pelvis. If not, it wouldn't. So this is a test for, again, this promoter. Is the promoter, if this fish does have the transcription factors it needs, if the complete promoter is wild type, that is, if it has no mutations in it that allows the transcription factor to combine, then you would produce, this fish would all of a sudden develop a pelvis. And this is another way to test for, that last experiment was testing the necessity. This one is a sufficiency experiment. Move the gene to a different fish. Is it sufficient to allow that fish to produce a pelvis? If so, it suggests that what this fish doesn't have, this injectee, what it doesn't have is a good promoter. By adding this promoter, then you get PIDX1 expression. That's testing the sufficiency of the complete PIDX1 promoter to produce pelvis. Fortunately, the last story is pretty short. I just want to introduce this. I could spend a lot of time talking about it, but you don't want me to, probably, and we're almost out of time anyway. Molecular evolution. So how molecular evolutionary biologists can tell if natural selection is actually acting on a gene. I told you a brief story about how there may be some selective forces in these freshwater lakes that might select for loss of pelvic spines. So if PIDX1 really is the gene that controls pelvic spine development, presence or absence, then we should be able to tell if natural selection has been acting on that gene, and here's how. A few slides. So let's think that this is an example of 5 different chromosomes from 5 different individuals in a population. So individuals 1, 2, 3, 4, and 5. And let's say one of those individuals, by chance, happens to have that beneficial mutation, the one in black. So let's say that's the PIDX1, whatever type of mutation it is, coding region, transcription factor, whatever mutation it is that allows that individual to produce a smaller or lack of pelvic spines. That individual is going to be selected for, and all of these other individuals that don't have that mutation will be at a disadvantage. They'll be killed before they reproduce, or they'll reproduce less, or one of their kids will be killed before they reproduce, or something like that. So what's going to happen is, I've also diagrammed these blue neutral mutations. That is, they're just random genetic differences between individuals, like the genetic differences between all of us. So there's some amount of genetic diversity in the population, but what's going to happen if there's really, really, really strong selection for individuals that have that mutation? What's the next generation of fish going to look like in terms of their genotypes? Are there going to be more of some and less of others? Which chromosomes are going to go extinct faster?
The neutral ones.
The ones that don't have the beneficial mutation. Maybe some of these individuals, 2, 3, 4, or 5, get killed before the next generation occurs, in which case there will be an increase in the number of chromosomes in the next generation or in subsequent generations. There will be a larger fraction of the chromosomes will have that beneficial mutation, because those individuals survive better and reproduce better. They have more kids that survive to reproduction on their own. And so as that chromosome spreads through the population over a short period of generations, what happens to all the blue alleles, the blue mutations? They're selected out only because they lack that beneficial mutation. So the point, the upshot is that if a gene undergoes natural selection that's really strong natural selection, like if you have this mutation, you survive, and if you don't, you die, that would be the strongest form of natural selection, which nothing ever gets that strong. But what happens is that mutation spreads quickly through the population. Generation upon generation, that one mutation goes to more and more individuals in the population pretty quickly, short number of generations. And as it does so, this is called a selective sweep. This mutation sweeps through the population, and as that happens, the individuals that didn't have it die, which means all of that genetic variation, all those blue asterisks, quickly disappear. So if you go into a population of animals and you genotype them and you don't see very much genetic diversity, that's a hint that one of two things has happened. Either there's been a recent really strong selective sweep, as I've described here. There are two choices, that's one. The other possibility is that there was a founder effect, which would mean like two fish got put into a lake and they had a bunch of kids, so the kids would by definition be very genetically similar to each other because they were siblings, and then they mate with each other and they mate with each other. So it's an inbred population, which also lacks genetic diversity. So this is one way to look for the possibility of strong natural selection, loss of genetic diversity from a population, or absence of it. The other type of test, so that's what this says. That's a selective sweep. No genetic diversity. There used to be some, but you go and sample in a lake, you see very little genetic diversity. Two choices, either selective sweep or a founder effect. There's a population that started from a really small number of individuals. And this is the last slide, of course. This is a numerical value that the authors calculate based on genotyping that I just described, about looking at the amount of genetic diversity in the population when they look at it. And all you need to know is the numbers. If they calculate an H value greater than 0, that means there's a deficit of alleles, deficit of diversity. And if there's an excess of high frequency alleles, that is, if there's a lot of new mutations in the population, not old ones, that's evidence for that selective sweep or positive selection. So what we're looking for is if they find the region of the genome that controls loss of pelvis, if it happened recently and was subject to that strong natural selection, we should see an H less than 0. That's it. I hope that's helpful, and I will definitely look forward to seeing you next week. Monday, Wednesday, 2 different papers.